Hi. So good afternoon, everyone. I'm Dr. Jacqueline Chung, and I'm very happy here to be the moderator for this reproductive health webinar. We're very happy to we have invited Dr. Fabio Cruz all the way from Spain to give this lecture on navigating patients' need, optimizing the Playbook in a real-world setting. So Dr. Fabio Cruz is a senior gynecologist at IVI Valencia for the last 16 years and professor at the University of Valencia. Dr. Cruz received his medical and specialist degree from the Federal University of Bahia at Brazil, where he also worked as an associate professor. After moving to Spain, Dr. Cruz completed a medical reproductive medicine fellowship at IVI, and after that, he obtained the master degree in human reproduction and later in advanced gynecological endoscopic surgery, both at the University of Valencia. So he's also currently finishing up a PhD degree at the same university. And Dr. Cruz has been a board member of the research committee at IVI and is also a part of the IVI-RMA Global Education as a professor and coordinator of medical training. So let's welcome Dr. Cruz.
Thank you so much for the opportunity to be here sharing my experience with you, and I would like to receive it the other way around. So I want to hear your experience as well. I want to learn from you and to exchange our practices. Right, I was initiating the presentation, and I want to be participative, right? So it's not a lecture; it's just a meeting. So you can stop me for ask whatever you want in any moment, right? I'll try to be brief. There is a special large number of slides, but I will go through them quite fast. And I have clinical cases in the end to make examples of the practice. Meanwhile, we don't need to go through about all that. That's being already kindly presented. I just want to say they have no conflicts of interest in this talk, regardless of the different fees from the pharmacological companies. And the only not conflict of interest about it, but it's the story that I have behind the Playbook Delta. I was in the Playbook Delta phase 2 study when we were determining the dose and selecting the algorithm. The phase 3, the phase 4, the launch of this molecule in more than 10 countries in the world. So I have some background, but it's not conflict of interest. It's more experience about it, right? So just quickly go through the beginning of this molecule. It was created to deliver a special creation of a molecule. I will talk about its principles, the development, results, and what is important here today, the clinical practice. So the principles of the Playbook Delta is its innovation, personalization, and the use of biomarkers. The innovation is because it's the first recombinant FSH, which is derived in human cells. Before that, we had recombinant phyllotropics, and we had at the same time human ones, but derived by urine. So now we have both of them together. It was created with the idea to personalize for the patient needs since the very beginning. Something very relevant about this development is I have never put some interest in the weight of my patients until I started that research. So yeah, it's true that we all believe that weight matters. So when we give paracetamol for a fever for our kids, we do it according to its weight. So if the kid is 20 kilograms or 30 kilograms, it will vary the dose of the medication that you're giving. But why won't you do this to the fertility patients? So, and before that study and research, I would only look at the patient and say, okay, the weight seems fine, or she seems overweight, but I never was concerned about low weight. If you look at that study, you can see that the same dose of reproductive were administered for every patient, some of them with 75 kilos, 50 and 100. So the same 20 kilos more or less would make a difference. Of course, it makes a difference in the blood levels of FSH, but the difference between normal and low weight, it's bigger than the difference between normal and high weight. That's the same 25 kilos difference, but it makes more importance. It makes its impact is bigger for the patients with a lower weight. And if I'm not mistaken, here in Asia, the patients usually they look more like this one here. In the USA, like here, in Europe, in the middle. So we maybe we need less medication for patients with a lower weight, and that can be measured, that can be correctly addressed. The biomarkers that we started to use, this idea is about 15 years old, right? The idea of creating the follicular delta. So back in the day, we used antimalarian, but not that much, but it was like inverting in the future. Nowadays, fortunately, we use it a lot. So the development was all about targeting the response to the ideal oocyte yield. What is the ideal oocyte yield? Well, 15 years ago, the idea was to have between 8 and 14 oocytes, because less than 8 is lower response, more than 14 could lead to high response and risk of OHSS, ovarian hyperstimulation syndrome. And 15 years ago, the classic way to make an embryo transfer was a fresh embryo transfer, which still exists, but now we use a lot of frozen cycles as well. So that was the phase 3 study that was conducted in many countries, and I had the pleasure to be a part of it as investigator. And the results of them were published in many, many studies. It is too much, so I'll go straight to the most important finding, right? First question is this algorithm that comes with the follicular delta can really target the response? And the answer is yes, it can really target the response, because that's the comparison between how many patients was meeting expectations with normal response. Not very high, not very low, right? So the blue line is the follicular delta, and the gray line is the follicular alpha. Those, according to our usual way of prescribing. In that phase 3 study, I see my patient, I was blind about the group, I was blind about the dose. I only see my patient in ultrasound and say, okay, she needs to be increased the dose by 50%, by 20%. And the nurse will adapt the dose if she was in the gray group. If she was in the blue group of the delta, it was the same dose that the algorithm determined since the beginning. And we can see that we have less patients having low response with the algorithm and less patients having hyperstimulation with the algorithm. So it was superior than all investigators in the multi-centric study criteria. We have criteria, right, to lower the dose, to increase the dose, but the algorithm was superior. Unfortunately, we believe we were superior, but no. So it can control better the simulation. Right? What about pregnancy rates? Well, it doesn't serve for nothing to target ovarian response if they don't get pregnant. So they should get pregnant, and yes, it works. The results were coincidental. It was empowered to demonstrate non-inferiority. It was not designed to believe that it's going to be superior. So it's at least not inferior. It's the same. What about frozen embryos? Because we only have one embryo to transfer. At the first, it should be the performance should be confirmed in the frozen embryos, and it does. So actually, it's the cumulative birth rates. So after all the embryos of the cohort, the results were coincidental as well. So it's not inferior in fresh, not in frozen. What about safety? The safety of this molecule, binded with the algorithm, was since the beginning very superior. So the probabilities to develop ovarian hyperstimulation syndrome with the follicular delta using the algorithm are half the possibilities of the classic way of using the alpha. And for those who doesn't know that study, the alpha was used with only 150 units. And then we can increase or decrease according to our judgment, but it's not a high dose, and even though the delta was superior in avoiding hyperstimulation. So then we came to replicate those findings. In Asia, usually researchers claim that their patients might be a different reaction to the gonadotropins, especially due to FSH receptors. They may have different polymorphisms. So it was replicated in a study in Japan, in another bigger study in Asia with more than 1,000 patients screened and randomized. It was made in China, Korea, Taiwan, and Vietnam. and it's more than 1,000 patients. And these results were very similar to the first study, with a difference that for the first time in the history of the Fabio delta, the results were not only non-inferior. The results for the first time came superior. And it was interesting to know, is it only a coincidence, or it's because the number of patients included were bigger, or because of the phenotype of the Asian patients are more compatible to the delta than to the alpha? So we don't know. It was the first finding, and it was some years ago. I will continue showing more evolution about the publications we have. And then comes clinical practice. Well, we all know that we can make clinical practice like the one size fits all model with one medication for the same patients, and they will have the results that they will have. Some of them will have good results, some of them will have average results, some of them will have even side effects or no results. And there is the personalized way of prescribing. So we rely on biomarkers, and then we give the proper medication or the proper dose for each patient. So let's imagine that we're not gynecologists. We don't dedicate our lives to IVF. We are oncologists. Can each one of us believe in a theoretical world that we can see a patient for prescribing medication, clinical medication against cancer, and don't follow a protocol and don't follow an algorithm? We just look at the patient and say, okay, you look like you need cyclosporine and maybe let's say a dose. No, we cannot decide by ourselves. It's a very serious matter. It's all about algorithms, all about classification. We are not free to use the dose that we want. And why in reproductive medicine we are free to do it? Because it doesn't change if they will live or die, but maybe because we are a very young field and we still need more research to understand more details. Right, but that's the first time that we were obligated to use biomarkers to prescribe any drug. Was the case of the Fabio delta. That is, using in a simple way in a mobile phone or in the computer, you can enter two data, weight, and antimalarial hormone. Why only those two? Well, it wasn't the phase 2 studies, and we were assessing different parameters: FSH, inhibin B, antral follicle count, weight or body mass index. Weight is superior than body mass index, because in the end of the day, if I am 83 kilograms, I'm 83 kilograms. If a patient with 100, much shorter than me, have the same weight, we need the same dose, because it's the body distribution is according to the weight, not in the body mass index, right? And it delivers the dose that we need to start the patient. And that's what the idea of this algorithm, and it was built when the era of the fresh transfer was predominant. And it works very well. But what if I don't need this ideal number of oocytes? What if I don't want 10? 10 was the average number, but 10 to 11 was the average number of the oocytes we have with the algorithm. What if I don't want 10? I want 20, because my patient needs 20, because my patient has a poor quality of this the husband's sperm, and we know that the quality of the results will be impaired because of this. What if it's the second cycle and the first one was not very good because we had low response? What if this patient, I don't want to do an IVF, I want to do an insemination, an IUI, and I don't want 10 oocytes, I want only 2. So I could not use this strategy in the algorithm in the first 3 years, 2 years and a half of this molecule. I didn't know how to use outside the algorithm. It was not authorized to be used outside of the algorithm. But then we came with a different study that I also participated to develop, then to make a correlation, equivalence between the micrograms when it's for Fabio delta to the international units that we are used to do. So this study compared the oocyte number, the number of follicles, estradiol levels, to make a clinical correspondence. And this clinical correspondence says that if we want to give the power of 150 units, we need to give 10 micrograms of this Fabio delta. So it's easy to convert It's easy to convert. It's not very complicated. I remember a funny situation. I was talking about this specifically in the USA, and one of the expectants, they said, I'm sorry, I disagree. I don't believe it's very easy to transform. It makes my life very difficult. So every time I need to do this, I have to think about it. And I said to him, I made a joke. I said to him, "Oh, you're completely right. This morning, I was trying to make my everyday run. I was trying to run this morning, and I couldn't because here in the US, you run in miles, and I run in kilometers. So yeah, you're right. We cannot transform. And I couldn't buy anything because I buy in euros and you buy in dollars. So it's impossible to convert. Conversion is something so we doctors, we cannot know how to convert because it's difficult. No, of course not. It's quite easy. And if you look at the mass of the molecule, because the fluorine alpha has weight, has mass, right? So we can measure it. And 10 micrograms of the delta resembles 11 micrograms of the alpha. It seems that the molecule itself, it's a little bit more powerful, but it doesn't matter. What matters is the correlation between units and that's because when we think about the power of the medication, we think, I want to do 225 units. Okay, 15. I want to give 300 units maximum dose. So for us in Europe, 300 is the maximum dose that we use. And okay, 20. It's very easy to convert and to adapt. And finally, do you know why we use international units to measure the doses and not micrograms? Because if you talk about painkillers, we use micrograms, antibiotics, micrograms. Every kind of medication in the world, we use micrograms. And why we use international units for gonadotropins? Do you remember that? It's an old story that I was obligated to dig in to do one of the lectures, and it's quite interesting as well. In the beginning, there were only urinary gonadotropins. We cannot trust the mass of the molecule itself because it was very heterogeneous. So even today, we have highly purified HMG, which we can rely on the weight, but not the best, not 50 years ago, not 40 years ago. And they needed to make an experiment called stem and Foley experiment to measure the power of these molecules. So we know I wasn't there. I'm young enough to not be 50 years ago working with this, but they injected the gonadotropin in the mouse ovary, and the ovary would grow. And it depending on how many the volume of the growth of this ovary, they classified by units. They created a different units to measure the power of the gonadotropin. So it's an indirect way to measure that can even lead to some failure. It's an indirect way. Measuring by mass is way more correct. If you just think about retrospectively, so what is the last gonadotropin we have in the market? Follicular delta. It is measured in micrograms because it's more modern. But before that, do you remember what is the one that we had before that? Cortifolitropin alpha, the long-term acting FSH, which is measured in micrograms as well. And before that, we have the antagonist of GnRH, cetrorelix, ganirelix. It is measured in micrograms again. So it's a thing from the past, the international units. We still use it because it's inside our minds. We were used to, and inertia is a principle of physics, but it's very strong, right? But well, it doesn't need to be like this. It doesn't have a special meaning for that. So let's talk about real-world evidence, real-world data, which is the most modern way to quantify and to evaluate the results. And why is that? Because in randomized controlled trials, they are randomized, great, and they are controlled, sometimes to control. In randomized controlled trials, I'm talking about the ones I have participated, not only with the follicular delta, I was participating with some studies from Merck as well, to historic Preg of Aries, etc. So when we use these, we control environment, and we select patients. In every kind of RCT for the pharmacology comparison, we cannot include patients with endometriosis, but they exist in our clinical practice. We do not include patients with the age superior of 40 years old, but they exist in our clinical practice. We cannot include patients with more than 28 of body mass index. It's not even obesity, but they exist. Well, maybe less here than the USA, but they exist. We cannot include patients with PCOS, etc., etc., etc. So we are taking outside of these trials half of our patients, and the most difficult ones, actually. So, well, these kind of studies of real-world evidence, they reunite all the patients that did treatments for real, and we get to know the results. So it's a different way to validate what is research. Right? So the three first studies with the Playbook delta in real world, they were very chronologically, they were very close to the first RCTs. So the way we have used those studies was still the majority of them with the algorithm, with this dose deviation. It's a different dose from the algorithm, only in a small number of cases, with those adjustments in only a few patients, and using the combination with other drugs like HMG in very few patients. So those studies, they serve for us as an external validation of the performance of the Playbook delta used in real world patients. The way to use was very similar, but the patients were diverse, and the results were very similar. That's 10 of signs that what we have in the RCT, and in those studies, 11, 10, 11, so very, very similar. So what the conclusion is, okay, it works outside of a randomized control trial. Next step, we had that big study, and it's very big. If you look at the number here, we're talking about more than 100,000 simulations, right? 100,000 simulations doing in 6 years of experience in Germany. So every IVF made in Germany in those 6 years were taken into account for that publication, and looking for live birth weight, what it really matters. So how many babies are born? Because every other detail, it's an intermediate detail, right? And because of the after propensity score matching, sorry, it was displaced. 4,000 patients used the Playbook delta because, of course, 6 years of evolution, the Playbook delta didn't even exist in the previous years, right? So they did a propensity score matching, which is a mathematical formula, statistical formula to compensate both arms. So we cannot compare 100,000 with 4,000. So we compensate with two arms with the same number of patients and the same characteristics. So we had equivalence for that. And the result was kind of interesting because we see that the cumulative live birth rate, the probability to have a baby in the patients using the delta, for patients compensated by the same age, the same environment reserve, the same year of treatment, or cannot compare patients from this year, from patients 6 years ago, because maybe technology is evolving. But the same patients, they have more probabilities. So we can say that in the last 6 years in Germany, the patients treated with the delta versus patients treated with the alpha, they got more babies born. And treated indeed with a single drug. Here, the exclusion criteria was mixing medication. This is the performance of the medication alone. And I'm sorry, I'm talking about delta versus alpha, but actually it was delta versus alpha and beta together, right? Because they were considered the same line. And for the last study that we'll mention today is one study from the two studies, one from Canada and another from Brazil, because in those two different countries, so actually in the whole America, North and South America, they usually manage different dose. They use higher dose than we use in Europe, than we use in Asia. And they have noticed that the maximum dose that the algorithm gives to us it's only 12 micrograms, which is 180 units. And it's a joke for an American. It's a joke for a South American because they usually give 300 or 150. So they try to make studies to compare the use of the algorithm versus their usual practice, right? And they usually combine with some LH activity. HMG is the preferred combination that they use. So these two studies we have seen here, the Canadian study and the Brazilian study, they have used different combinations of the menopur using adding immunotropin to the 12, which is the maximum dose of the algorithm. So they don't disrespect the algorithm. They use the dose that the algorithm gives to us, and they give more HMG. If the patient is a low weight, less HMG. Normal weight, 150. In high weight, more than 100 kilos, so 225. And the Brazilian study was more easily, so 150 for everyone, because they don't have so many variety of weight there. They're usually normal weight. And the results were interesting. Sorry, before the results, the patients were different. They needed to give this dose and this combination because they needed, not because they wanted. Why is that? Well, age, the patients were older compared to the patients from the original studies with the algorithm. The patients were heavier, they had more weight, and the patient had lower ovarian response. So yes, they needed to compensate with this extra HMG for the patients older, heavier, and with lower ovarian response. And the results were that they have much more embryos in the end. Makes sense if you boost the response with more HMG. HMG is not only LH, HMG is FSH plus LH. So in the end of the day, you have more oocytes, more embryos, and probably more pregnancies. Both studies were designed to find embryos and not cumulative liability, because it takes maybe 2-3 years to take into account cumulative live birth rate. And they did it in only 1 year. That's why it was shorter. And for finalize this, I brought some clinical cases, but before starting them, I would like to ask if you have any questions and ask if you have any case sharing from your side later, because if you have, maybe I don't need to present mine. Do you have any?
We do have some cases, but not considered very brief, because this is just a general practice experience.
Okay. So I will show some of the cases. I don't want this to be very long, so I will be very brief in selecting some of these cases. The first case for a naive patient, for example, doesn't seem very interesting. It's a very simple case, right? Let's go. Well, let's start it very quickly. So 34 years old, 53 kilograms, male infertility. It was a simple case, but this was my first patient that I ever gave the Reproductive delta. So it was way long. I don't remember, 7 years ago, with a normal antimullerian range, normal follicular count. And I, instead of selecting myself the dose, I used the algorithm because I was used to use the algorithm during the studies, and I see people getting pregnant, and I trusted. The interesting thing is that the algorithm gave me 8.3 micrograms. In that day, I didn't know what 8.3 represented. I had a clue because I was inside of the study, but I didn't know for sure. I said, okay, 8.3, go on. Today, I know that this is inferior than 115. Anyways, it's like 110 or 100. It's very low dose, actually. I would never have given this dose to my patient, but it worked because I had 11 oocytes in the end. And the patient got pregnant and still have 2 more embryos frozen. So I would never imagine that such a low dose could give me such amazing results. I would definitely have given the double, just in case, just to be sure. We are a private clinic. The patient don't accept failure because we cannot give another attempt for free. Anyway, just quickly going. Let's high responder. They're all real cases, right? So this high responder, let's call her Susanna. 33 years old, 55 kilograms, and tubal factor as the cause of infertility. The anti-malaria was higher, 5.6, between 3 and 14 antral follicle count. So yes, there is a risk of hyperstimulation here. And this patient, she came in after an IVF failed in another center. They started with 150, and the response was very slow. It took some time to get recruitment, so they increased the dose to 225. And in the very end, 2 days before finishing, they got back to 150 because they believed she was responding too high. So maybe they should have been patient to wait without increasing the dose. Anyway, she got 31 oocytes, 4 blastocysts that they were frozen because a freeze-out strategy was performed, and she didn't get pregnant. And besides not getting pregnant, she was unlucky enough to have some of those embryos that didn't survive after the freezing. So we know that the embryo not surviving after freezing is not very often, not nowadays. This case is 7 years old. And I'm afraid that the quality of the lab of that clinic was not very good. But anyway, it is a real case. And the patient comes to me asking 2 very important tests. One, she wanted to transfer an embryo in a fresh cycle because she was afraid of freezing embryos. I told her that we have a good lab, that she should not fear, but she was afraid anyway. So I should try to make a transfer in fresh. And she said, and of course, I don't want to suffer a hyperstimulation because I had a mild hyperstimulation. It was no fun. I want to do both things. Well, it's hard enough because better results and lower hyperstimulation. So I went straight to the algorithm, and it was an amazing slow dose of only 6 micrograms. It's very close to 75. It's 85 or 90. It's very low dose. I didn't know back then. It's the better because if I knew, I would give more, right? But it worked with 13 mature oocytes, no HSS. She was triggered with HCG because we were planning for a fresh transfer. I was crossing my fingers and closing my eyes. I hope she doesn't hyperstimulate. And it was okay. It did well. It did well. So it's an interesting way to face patients with the risk of ovarian hyperstimulation. If we use the algorithm, don't worry, the dose, it will be correctly calculated. It will be low, but it works to maximize the possibilities to transfer in a fresh. Now comes really difficult patients, right? Those 2 patients were exceptional. I don't have them every day in my clinical practice. I usually have those. The first one, so it's not so interesting case. Let's go to the second one. Right. So Pilar, patient number 4, 63 kilograms, advanced maternal age, a previous cycle with low response, and 0.7 of anti-malaria. So I was seeing between 4 and 6 follicles every time I saw her in the beginning. And what is the right approach? Giving 450 doses for everyone, or mild stimulation like the northern countries in Europe, they usually give 100, 150 for low responders because they believe that she's not giving more than 4 or 5 oocytes. So let's give a low dose anyway. So what I did for that patient was using the algorithm because I was in the moment where we should respect the algorithm. Okay, it was not bad, but not good. It's only 3 oocytes, only 2 mature. That was really matters, and 1 aneuploid embryo. So yeah, so what we do here, there is nothing. So sorry, this patient has 42 years old. And in our setting, when the patient is more than 40 years old, we perform PGT, right? PGTA to avoid risk. It's not obligatory, but we offer the patient to do PGTA for this kind of age. And after this, we consider to maximize the dose for a dose that she really needed. So it's not too much if you took into account that 12 micrograms, the algorithm is 180, and going up to 20, it's 300 units, which is the maximum dose that ASHRAE, European Society of Human Reproduction and Ethnology, our society, they recommend. And with that approach, we moved from 3 oocytes, 2 mature, to 5 oocytes, 5 mature. And fortunately, she had more luck in that cycle. Right, so we can even, by increasing the dose, in that case, with especially for older patients, because we don't know how the follicular delta develops in older patients, because the studies, they were excluded. But now we know, but not only by my case, but because of the other studies, that older patients might need a little bit more dose. And the last case is another low responder. The difference from it's very similar to the previous one, right? The difference that she has, she's more heavy than the rest of the patients, 80 kilograms. So with this patient, I tried to use the algorithm because I was obsessed about the algorithm. I was studying the algorithm, helping creating the algorithm. So in my first years, I used for everyone, but it was canceled because there was only one follicle growing. I know she's a low responder, but well, only one is too low. Maybe we can have 3 or 4. And in the following cycle, it was just after the Canadian study was published to add HMG. So yes, she's a high weight. I added the maximum dose of the algorithm plus 150 of HMG of minotropin, and the result was quite better. So we got 4 mature, which was not perfect, but was the maximum that I could get from this patient. So nowadays, for patients over 40 years old, for patients with a high body weight, I wouldn't even try this. I wish since the beginning, start with this protocol combination because of those 2 factors, age and high weight. There is enough evidence in the literature and in clinical practice that I shouldn't use very low dose, like only 180 units for patients more than 40 and with a high weight. And that's the proposal of the study. You can read the study later, it's not too much information. So they propose for good responders only 75 of HMG, for poor responders 150, and super heavy poor responders 225 to compensate. It's not my authorship, it's from Dictionnaire Kadosh from Canada, right? And the final case, and I will finalize in only 1 minute and a half. This patient came here for performing PGT. It was a patient of only 35 years old, normal weight, but she has a dominant mutation, and half of the embryos will be abnormal. In our everyday practice, in our genetic lab, when we do PGT-M because of mutation, the technology that we use can be applied to PGTA in the same time. So as we can, let's do both of them together. So at least half of the embryos will be discarded, plus some that could be theoretically aneuploid, because every human being can create aneuploid embryos, right? So maybe just the minority, but so more than 50% of those embryos will be discarded after the PGT because of the problem. Right, so the ovarian reserve was quite nice. So 3.7 nanograms. It's higher than our average, and she could give a good response. If I stimulate that patient according to the algorithm will give me only 9. So because with a light patient with a young age, the algorithm, I calculated myself and I saw 9. And I said, no way. I don't want 10 oocytes for that patient because it might be enough, but it might not be enough. So I decided to go to the protocol that we use the most in our clinic, which is using 15 micrograms of the delta, which is 225, and using 75 units of minotropin. Why is that? Well, 15 micrograms of the delta, it's just a small tweak, right? We know that the pin of 72 don't have 72, they have more. So we can use for 5 days of 15. And the average duration of the stimulation is 10 days, right? So with, well, this case was 12, but the average duration is 10 days. And so with 2 pens, we complete one simulation. It's quite easy. And 1 extra ample of HMG every day. So we had in the end 24 mature. That was enough to have 7 embryos biopsy and 3 of them non-affected and euclid. If I had, instead of 27 oocytes, if I had 10, maybe I would have only 1 embryo. So this patient needed maximization of the protocol. I'm not hyperstimulating my patients just because I want. I was doing because she was in a specific need of that. And the trigger was with only GnRH, so OHSS free, and she had no problem at all. So in this kind of patients, we always do the freeze-all because she needs a freeze-all for the PGT. So we are free to stimulate a little bit more because we have no fear of hyperstimulation. The final remarks and take-home message is that the Fluidotropin delta has an algorithm that was validated and it works in targeting the ovarian response. It reduces the odds of ovarian hyperstimulation syndrome. There is an extra need. We cannot don't waste drug if we predict in advance the dose that the patient will receive. It is simple because the old algorithms, well, I hope you didn't work with the old algorithms because we needed to put a big table and use a lot of parameters like FSH, body mass index, follicle count, AMH, and now it's way simpler. And if we're not happy with 10 oocytes, we can convert the dose and search for 20, search for less if we have any questions. And more recently, and for the countries that embrace the idea of combining drugs, the combination of HMG and the Fluidotropin delta seems very promising. The 2 studies we have are quite recent, and I'm sure in the future we have more research pointing this direction. I confess that I am using more and more this last approach right now, but we'll see in the future more publications in that. And those are the final remarks. I want to thank you very much for your attention. I will open for questions right now, and if you well, I prepared some slides about the research because you said you're interested in research, and I was. But before that, we can talk about if you have any questions of the first part. The second part is quite brief, only 5 minutes.
So thank you for your presentation. So actually in our unit, we are following the latter approach where we usually add the we add manager during the simulation because it seems that initially we just used reproductive results, but it seems that the response wasn't as good as what we expected. So we had to add a little bit of manager in our patient and the response is better. So this is we also try to use the dose and conversion method because in Hong Kong, actually we are using higher dose for the patients. Use up the full 15.
Right. So, okay, no, it's good to know you are completely aligned to the tendency of the world to combine and to use higher dose. So we have in Europe they are usually very straight about the dose thing because of the society, etc. Sometimes we use more dose, especially if the patient is heavier. Well, it's not a rule, it's not forbidden, but it's a recommendation. But yes, sometimes we can use a little bit more than 300. I personally have 300 as the maximum, but if she's overrated, we can go up to 450 as well. So it makes total sense. And why is that? Because the publications from the algorithm came from ideal patients. Good responders, young. So how can it be wrong? So so if you receive in our unit young patients with no problem, only problem with the sperm or with the tubes, and they are good responders, we can rely on the algorithm there's no problem. It's a beautiful thing. We inverted many years in research, and I still like it. But for more difficult patients, definitely that's the way to do it. And I'm happy to see that we agree on that.
So
Thank you for the wonderful talk. So I guess in our usual practice, the patients where we use recombinant FSH are those who are usually like older, higher body weight, and those poor responders. Yeah, so I would like to ask, do you have any experience in using recombinant with recombinant LH? I know the literature is most of them, they just really like specify they use the LH from HMG, or it's like a recombinant LH. Or do you have any other sorts of experience? Because for us, if sometimes for those especially poor responders, we use GONOF and recombinant LH sometimes. So just want to ask whether you have any experience in that. Thank you.
Very good question indeed. We don't have experience with mixing both of them because of three reasons, right? Reason number one, all the research that we have combining the delta is combining with HMG, which gives LH driven power. Second reason, in Spain, in all the countries that I know, it's not possible anymore to buy recombinant LH, only in combination with recombinant LH. So talking about brain names, we don't have blueberries anymore. We have only berries. So it's complicated. We cannot take the FSH from the berries to add the delta. So it makes it impossible in clinical practice to mix them both. And third, even when we had the recombinant LH, it was in the old days where it was, it felt like unnatural to mix medication from pharmacies. So we always, it felt rare, it felt weird. And we usually do this with the same, because they should respect the same compounds, etc. I don't know. But anyway, the third reason is less important. But nowadays, even if I wanted, how can I do it? So I have to use, which is the mixture of the combined, it's 150 plus 75. So I will give this more 5 of the regular delta, it would be like odd, right? So that's why it makes it a little bit more difficult. And I'm afraid we cannot research that, right? But it's the evidence that I've been talking about. The evidence that is published is which LH activity from the HMG that is supposed to be similar.
Thank you for the wonderful talk. I would like to ask for a special group of patient PCOS for cystic ovarian syndrome. With this said, your stimulation pattern is the same because they already incorporated in the AMH, or you have other experience, particular for this group of patients. We do have some problem in occasionally using a conventional stimulation regime for PCOS.
Right. In this case, I have to be humble enough to ask to learn from you, because we have very little experience with PCOS. First of all, because in our setting in Europe, there are not very prevalent phenotype. And second, because in Spain, we have very good public hospitals that covers up to 3 cycles. And all the PCOS patients, they have criteria to go to an IVF in a public hospital. And usually they are good prognosis patients because we have a lot of ads. So hardly I receive in my private unit a patient with PCOS, because they usually get pregnant in the public hospital. So I have very few cases, and we didn't build a lot of research nor experience in those cases. I had some, of course, because sometimes they don't want to wait the waiting list. But the majority of them, they have more data, more research, more experience in our private hospitals, which I'm not part of them. So I would like to ask you, how do you stimulate those patients?
Usually use lower dose, but the problem is, as you say, well, they may respond with a very high responder, but they also may respond without any, dominant X. Okay, so it's a bit difficult. I think one factor, of course, is the body weight. And then, okay, and well, this is a difficult group of patients. But we seldom have experience using, the Reproductive in the PCOS.
Right, I have no personal experience, but you just mentioned that patients with different weight, some of them, some types of PCOS, they are associated with obesity. We're talking about patients with a very high insulin level that we don't see every day. So it seems that the algorithm can help us. We don't need to follow the algorithm like religiously, but it can be a guide for us. Once I was in Russia making the talk about the algorithm, and one of the guys said, "Oh, can I say the algorithm is like a guide? It's like, I have an automatic car and I can put it in automatic, but if I want, I can take the automatic and do my own way. Of course you can. So it's not obligatory. You can calculate, okay, the algorithm says that I should use 6. Oh my God, 6 is too low. I would use 150. What is in between? So let's use 8. Well, let's make some changes, and it's a guide for us. Maybe so for hyperstimulation, we should be here. If you don't mind freezing out, we can simulate the way we want. So I don't have the answer. I don't have the expertise, but it makes sense to have the algorithm on our side, just at least to consult, not to follow 100%.
So can I just ask, if there are any, or during your experience, any difference in the medication in different protocols, for example, antagonists or PPOS, is it what the same or? It's one.
In our experience, the results are pretty much the same, but with the classic long protocol we don't use anymore, and with the PPOS, usually we need LH activity more because the patient is more depleted from endogenous LH. So we do a lot of PPOS. We do a lot of freeze-all because of PGT, because of our type of patients. And with the PPOS, we usually give them some dose. Let me just, okay, too much. Usually we give the smallest dose of HMG to cover the probable lack of LH that you might have. So it's not, we haven't published anything about it, but in our experience, for these protocols, we need more LH than the antagonist protocol. But well, the studies say as well that the older the patient, the more they need LH. So we always, not always 100%, but the majority of our case, we give some period, just to reassure we're not losing these receptors.
So, are there any questions from the floor? What about the laboratories? Are the embryos better in quality? Do you have more good quality?
That's a very interesting question. There is a lot of data. I couldn't bring everything, otherwise I would be 4 hours talking, and it was too much. But there is some evidence that in Japanese study that was just 5 days ago in the Aspire meeting, and that was Dr. Kobanawa was bringing his results from a big study in Japan with the delta. They have a little bit less oocytes, but better pregnancies. So he was thinking about maybe the quality is better, because it's difficult to say the quality is better, because it could be the opinion from top. So we need to see if the babies were born. Right, so the live birth in his study was a Japanese study, recently published. It was superior with the delta, but they didn't have more oocytes. And even in I don't have the slides, but this German study with 4,000 patients each arm, the everyday dose of the delta was about 15. Oh, sorry, no, it was about 10 point something, so about 150, 160 maybe. And the average dose of the alpha and the beta was 190. So less dose and more babies born. So it is pointing towards the direction that there is some changes in the embryo, but it's too soon to say this so quickly, right? So we need more research on that. But there is a tendency that maybe in the future we can see. Nowadays, it's just a small tendency.
So, any other questions on the floor? If not, and would you want to share the research?
Yes, right. It's more conceptual than specific, but well, we are partly for those that don't know our group, EV-RNA. So initially we're EV means Instituto Valenciano de Fertilidad. It's an institute from Valencia. Valencia is the city that which I live, and where this was founded. It was created 35 years ago. In April, it completed just right now 35 years of history. And the first clinic was in Valencia and started to be open in other different cities in Spain and outside of Spain, Portugal, in Italy, in the UK, in South America as well. I'm South American, I'm Brazilian, so I'm there for the last 17 years because I was trained to open a clinic in Brazil, and things go different ways, and I stayed forever in Valencia, and in Chile, in Argentina, Mexico, etc. And like maybe 10 years ago, we made a fusion with Reproductive Medicine Associates, which is a chain of clinics in New York, New Jersey, USA. They were the best clinic in the USA, not the biggest, but they had very good results in research. So we decided to be one sole group, and it was 10 years ago. Nowadays, maybe 2 or 3, I don't remember the dates precisely, but maybe 2 years ago, the group was incorporated in a big portfolio. So big investors have bought the group and make more acquisitions. So there are very relevant groups of clinics around the globe that they are part of what we call global research. So we have now the clinics from the North Scandinavian countries. We call Livio Group, which is more than 7 clinics in Sweden, Norway, and Iceland. There is a big group called Genera Life from Italy. You must know Filippo Baldi and all the big researchers. They are part of the group as well. So it became much bigger, and we always did research, but now we do in a different way. Now we do in what we call the EV innovation. So we're always trying to make something new. It's a very bigger thing than just me, of course. I'm very small compared to the whole group, but we always trying to find innovation in what we call our research alliance. So it's not in the beginning it was Valencia researching, and the other clinics replicating our research. And nowadays, every clinic has their own, not every day, from 5-10 years ago, every clinic has their own resource to make research. And there is a central mechanism, which is called, I only say the name in Spanish, but it's a organ inside of us that regulates the research and helps. Because, for example, I like research, but I'm not very good writing the research protocol. But they have people to help me write the study protocol. They have ethics committee. You have to submit the ethics committee. We have a team that help us present to the ethics committee and make our life easier. In the same way, they make our life difficult because we cannot just research whatever we want. Okay, we research that. Why? We have another research similar like this in our clinic in Lisbon, so we don't do it. And so it's good and it's not good for many reasons, but of course it's the better for the whole group, right? So just yesterday, we are thinking about making a specific research, and they say, there's no need. We are already doing one of this in another clinic from the same group, so we don't need to do that. Okay, but in the majority of the cases, it's for the better. And the third branch is what we call EVR Global Education. So we export our results. It's less of research and more about training. But we still do research and we still stimulate research with EVO education. We receive regularly, so it's an email that I received for the first 2025 quarter, so the first 3 months of the year, with all the publications from the group, and it's so I couldn't put all of them. 43 is the last one. There were 43 different publications from the group, and it's a lot. So some of them are from our group, Castells, so they're from our clinic in Valencia, but some are not. So we get lost. We need to be informed about what we are producing because we cannot track anymore. It is open in our website. If you go to EVRMA Global Research Alliance online, we can, they offer our research. Some of them are open access and we can enter there and we can download, we can follow, we can study as well. We celebrate scientific congresses. This was just last week, it was so maybe 10 days ago. I was there in Barcelona. I was presenting. I was part of this congress, and it's not only a congress to discuss. It's a congress where we stimulate science and research. We even give research awards. In this case, Nick Macklin, you might know him. He was the winner of one of the prizes that we have given, and it's it's to recognize, is to acknowledge the researchers not only inside of our teams, right? So Nick, for example, is not in our team, but he is an outstanding efforts for research, and we award him as well. And we always try to make science accessible, and so we have in our web some open access, some them for just for everybody that don't need to be part of the group to have access of them. Of course, being part of the group, I have access of all of them, even if they are not open access, right? And finally, I dedicate a lot of my personal efforts in EV Global Education, which is a platform for medical training. So we receive fellows from the training. I'm now receiving a fellow from Saudi Arabia. He's with me for 1 year. He's been already for 8 months, so more than half of his formation. But I received fellows from Brazil, from Canada, from Italy, from France. I don't even remember because it's been very frequently. So there are online courses. There are this is our direction of research. So we sometimes publish like the experts' view. It's all in Spanish because it's the resource that I have, but they have in both language. And now they are exporting for even more language rather than English, right? And there are a lot of online courses, some presential. Of course, in embryology, they should be presential because they should see some details. And some clinical courses, they are online. I'm coordinator and creator of what we call the advanced simulation course, because we have the basic and we have the advanced, and it's 100% online. We're now doing the new version because it was launched in 2021. So 4 years later, we should adapt to the new publications, etc. So if you think about doing the course, don't do it right now. Wait for the beginning of next year because it will be revisited. And these are some of research that we're taking, that we have conducted in our own center. So I can show them, I can talk about it because I was part of them. Some of them were not completely mine, but I was involved. So recently we have investigated and published a lot in the PPOS protocols. We do quite a lot. So for 100% of our egg donors, it's PPOS. We have a lot of egg donors. We do a lot of egg donation. It's the country in the world that makes more egg donation to Spain, not only for local patients, but for foreign patients that goes there. All of the fertility preservation, because we now in the private center, we have a lot of fertility preservation. We do a PPOS as well, because both of them need no transfer, and they can get benefited by using oral medication, cheaper medication, because it's all out of pocket for the patient. There's no reimbursement in our clinic. And there is one personal project, which is an epigenetic signature in the sperm for the possibility to develop autism. For those who don't know, I have a son with autism, and I dedicate some part of the research about this. It's still very initial, that there is nothing relevant to use in clinical practice, but we keep researching, right? And our director in Valencia, Ernesto Bosch, his main field of research is to personalize ovarian stimulation. So I would say the patient-tailored infertility care is his biggest leading research arm. He's one of the consultants, maybe the main consultant from the ESHRE, which is the American Society, the European Society for Human Reproduction in ovarian stimulation. So if you download ESHRE guideline for ovarian stimulation, the first author will be him. So it's his, that's what he usually researches and his main expertise. And besides that, you might have seen that Elena Navarta is one of our colleagues that publishes the majority of the publications in progesterone in artificial cycles. But for embryo transfer, the levels of progesterone, the day of the embryo transfer, so it was an original research from our own. Nowadays, everybody uses the measurement of progesterone. And we keep researching, not only the first one, it was 2020, there was almost part of it as well. So, but we keep researching to go to the next level. And that's pretty much what I wanted to share with you. I thought for the ongoing research we cannot share. It's something that we need to wait, and encourage you to research or to follow closely the newest updates. And again, thank you for your attention.